Okay, so welcome back to our final session, which is kind of our translational session with some quizzes and some more QA. So at the end, we encourage you to hang around if you like to have some chats about setting up neurofeedback or any questions you might want for your group. So, but now we're going to have a presentation from Ethan Murphy. Ethan's a final year PhD student at ACU, and his project can be separated into two streams using functional magnetic MRI techniques to either map or mitigate the brain in people with cannabis use disorder. In particular, his PhD project aims to be the first of its kind internationally to use real-time FMRI neurofeedback to decrease craving-related brain activity in cannabis use disorder. Separately, he investigates countries' sustainable development goal performance and supervises master students in resting state functional connectivity. So thanks, Ethan.
Thanks, Beck. Okay, so I kind of like this image because anybody that's been a PhD student knows this is kind of how you feel when you start off your project. You feel very much like a hitchhiker on the side of the road. You don't really know what's going on, or you don't really know what you're supposed to be doing. But luckily, there's a car coming down the road of some well-established researchers who can really show you the way. So one of the things with hitchhiking is that you should really know where you're going. That's probably a good idea before you get in the car. And one way we can do that in research is through a systematic review. So I'd recommend anybody trying to start a project, particularly in neurofeedback, to really get a good idea of the literature before you start developing any paradigms, before you start developing your methodology. So I'm going to talk a bit about my systematic review and how we used it to not only tell us what had been done in terms of the literature, but potential avenues that we could explore. Cool. So the objectives of the systematic review were threefold. The first was really to characterize any neurofeedback-related neurobehavioral changes, so any brain activity or subjective craving changes that were found in any substance-using cohort. The second was to try and investigate if there had been any association between these two, so if there's any correlations between the potential brain activity changes and changes in behavior. And the last one, which is probably more relevant to the actual project that we were going to develop, was to assess the methodological quality of the literature, and that's what I'm going to spend a bit more time talking about today. In terms of methodology, pretty straightforward, pretty boring systematic review, followed the PRISMA checklist. So this was started back in March 2023 when I'd just been a couple of months as a PhD student. Finished up the searches, particularly thereafter. Really the effects that we were looking at were pre to post neurofeedback, so at the start of the session compared to the end. Also between conditions, so if anybody had actually used placebo control conditions to investigate any changes in activity, brain activity, or craving between those two conditions, and then if there was any condition by time effects. So in terms of what we found, so this was up until 2024, and I don't think there has been any new studies since then. But to date, there's been about 16 studies in substance using cohorts, so still a relatively novel field. Most of those tend to focus on three main substance using cohorts, so alcohol, tobacco, and there was one in cocaine. So what that means is the generalizability for us, who's looking to investigate in cannabis users specifically, is a bit limited, but nonetheless we can just work with the research that's there. In terms of what we found then of results, and this is beginning to lead us into the whole idea about developing your methodology based on the literature. The first one, A, on this figure here, represents the targets that were used. So the majority of the literature used an ROI-based approach, so not the functional connectivity or other approaches that the guys have spoke about previously. Relatively straightforward, an ROI was picked. The size of the spheres in this figure from the systematic review represents the number of times that a study had used that target. So you can see the ACC was coming out as a major target for any substance using cohort, and that is in line with our non-neurofeedback literature. We know that the ACC is involved in things like craving regulation, but also emotion regulation. Also other reward-related regions like the striatum. Again, prefrontal regions like the DALPFC, insula as well, sort of tapping into those emotion regulation, craving regulation regions. In terms of the actual results, the results are promising but early. So we did see some pre to post changes. So before and after neurofeedback, people were seeing changes in those regions. So in the SEC, for example, in the insula, in the striatum. Again, in terms of between condition effects, there is between condition effects, but a lot of these are between a neurofeedback and a non-neurofeedback condition, which is not a great placebo. So a lot of these between condition effects don't actually represent placebo-controlled effects. So they have to be considered in light of that limitation. And in terms of condition by time effects, so this is really between the neurofeedback and a non-neurofeedback condition, over time, a lot of people weren't really seeing any changes. And I can get into a bit of why that might have been the case in terms of the limitations of the literature. But arguably more relevance for anybody here that's looking to develop their own neurofeedback protocol are these limitations and recommendations from the literature. So as I mentioned, most of the studies were not placebo-controlled. And the guys have spoke about previously that an active control is probably the best control condition to use. There's still a lot of discussion about what that looks like, whether it's from a different run, perhaps from neurofeedback, whether it's from a different participant, or whether it's from a different region. The one thing that I would recommend is that if you are going to use a different region, you need to be sure that it's not involved in either your task, so craving regulation, or neurofeedback itself. So in Sampers's slides, he did cite there is a really useful meta-analysis that talks about regions that are involved in neurofeedback itself. So there was studies in substance using cohorts that thought they were using a control region. They thought they were using a region not involved in the process. But it turned out this actually changed with neurofeedback. So in terms of picking your control condition, a different region is a pretty valuable condition to use, but you need to be sure that that is actually a control condition. So you can do it with the literature or with any pilot runs as well. The other limitation is that a lot of people don't actually include a transfer run. So I think somebody had mentioned in previous presentations what a transfer run is. Basically, it's after your whole neurofeedback task has been completed, you use the same task, except you don't present any neurofeedback. So what you're trying to investigate is if participants can use the strategies learned during neurofeedback, if they can use that without being presented that information. And that's really where some of the clinically relevant information here is, because as has been mentioned, like these scans are very expensive, they take a lot of time and a lot of resources. But if we can investigate if people can actually learn to use the same strategies outside of a neurofeedback context, that's really where the goal is from a clinically relevant perspective for neurofeedback. Likewise, from a bit of a clinical relevance standpoint, the literature was severely limited by the lack of behavioral follow-ups. So again, one of the things with neurofeedback that makes it different from other neuromodulation techniques is that a lot of it, your stimulation is internally driven. So what that means is participants' strategies matter, and what they use to generate that neurofeedback signal really matters. But it also has implications for the dose effect of your neurofeedback. In a sense, participants could learn the most effective strategy only during one session, and if they then practice those strategies at home, like through some app like Sampers has used in his design, or some like homework type of assessment, you essentially have an almost infinite supply of dose, because participants can just practice these strategies at home as often as they want. So I think that's really important as well, to try and establish a framework so that when they leave the scanner, participants can really drive down the strategies that they've used and practice those at home, try to increase that dose effect of the strategies. Again, some of the big limitations as well. Most of the studies have very small sample sizes, so we see generally between like 10 to 15, whereas most of the literature is pointing towards at least a sample size of 20 to see medium and large, or medium and small behavioral and neural effects. So again, it comes down to feasibility. A lot of times you might just be running a feasibility study, so 10 participants can be, like, that's sufficient. But if you are looking for more like RCT, those type of bigger studies, I would recommend using more than 20 participants if possible. One of the other big limitations as well is the reporting of the variables. So again, people do all these different, like really nice neurofeedback designs, but they don't really tell us some key variables. And one of the key things is strategies. So during the task, what strategies are people actually using to change their brain activity? That's a key one. There are some other ones, and it's very useful to follow some of the already established guidelines, like the CRED-NF from Ross et al. So anybody that is going to publish the results, I think at that stage you need to check the guidelines and the best checklists. And the other limitation refers to more about substance use itself. So a lot of what we see in the neurofeedback research is that if they are characterizing the neurofeedback study and the neurofeedback design, they don't tend to characterize the participants that they have. So they can classify, for example, alcohol users as just alcohol users. They're not specifying how severe of an alcohol use disorder they might have, or how much alcohol they might use, for example. And these things could all impact the degree to which they can regulate their brain activity in neurofeedback. So yeah, good to characterize that as well. So yeah, in terms of final thoughts, the evidence in substance use disorders is promising, but largely inconsistent. There are some key mechanistic and methodological understanding that's still required. So some of the things that I mentioned, like a transfer run, a placebo-controlled condition, those things are all still needed so that we can be confident about the results that we're seeing. Yeah, and that's kind of where our study aims to come in. So it aims to be the first to look at neurofeedback in cannabis use disorder specifically, but while trying to address some of these limitations. Yeah, and the good news we actually found out this morning, the protocol for our study has been accepted for publication, which was a nice thing to wake up to this morning. But yeah, so you can scan the QR code, keep up to date with anything that we come out, and hopefully we address some of these key limitations. Just a thank you to all the team that's here today and the people that couldn't make it, unfortunately. None of this would be possible without any of them, so thanks everyone, and thanks to the funding bodies as well.